So what I'm going to talk about today is self-organization of charged macromolecules, particularly of intrinsically disordered proteins and polysubstitutions. So part of the talk might not be directly about biomolecular systems, but still it is relevant for biomolecular condensation. So to a physicist like me, the biological cell is like a Coulomb soup because all the typical cellular compartments or organelles constitutes of various types of charged molecules and also electrolyte ions, and that's why the fundamental forces that are operative in this dense crowded cellular environment arise from the action between different charged components and also from the weak range van der Waals type interaction through which the different components communicate or interact with each other. And how does this communication and cooperation happens, and what are the factors that that modulates this communication, that is of fundamental interest to us. Now, the speakers before me have already, like in great detail, introduced what biocondensates are, so I won't go into much detailed introduction about them. So we know that biocondensates are basically hierarchical structures of different types of proteins and nucleic acids, and in our study we were particularly interested in the biocondensates of intrinsically disordered proteins, which are known to self-assemble into membraneless organelles or droplets through liquid-liquid phase separation. Now, but overall, the phase behavior of this IDPs can be quite complex because of the confluence of many different attributes. For example, there are entropic contribution due to the chain connectivity of these macromolecules, and then we have electrostatic monopole and dipolar interaction, and also hydrophobic interactions between the different segments. And even the overall sequence of these macromolecules are expected to have an effect on their overall phase behavior. So to explore along this idea, we considered the particular IDP alpha-synuclein, and then investigated its aggregation mechanism. So this here you can see is the sequence of this protein. So if you look closely, so this is the N-terminal segment, which has a net slightly positive net positive charge, but if you look closely, you will see that there are many pairs of positive and negatively charged residues in this segment, and that's why this segment has an overall dipolar property. And then there is this intermediate NAC region, where you will typically see the different secondary beta structures. And finally, there is this C-terminal, which has a very high net negative charge. And because of the concentrated charges at the termini and the overall hydrophobic center, this protein typically shows the characteristics of an IDP. Now, this is actually a well characterized protein in experiments, and what we know from experiments is that in bare solutions of the protein, droplets are very hard to form, but when you have crowded molecules like PEG, you will see that droplets or membraneless or biocondensates very readily form in the solution of this protein, and these droplets over time can transform into more viscoelastic and solid like amyloid fibrils, which are believed to be connected to the neurodegenerative disorder Parkinson's disease. Now, in our study, we did a detailed detailed investigation of the structural organization in the solution of this protein using coarse-grained simulation, and that's what I'm going to talk about today first, and then later I will show some extension of this study to how does complexation with RNA affect this structural organization. And finally, I will also talk about one important aspect of not just this IDPs but other charged macromolecular systems, which is the effect of sequence. So, so as I said that we basically do a coarse-grained simulation, and the coarse graining is at the level of one residue, at the level of the residue, and obviously we have the aspartic acid and glutamic acid, which are negatively charged, and lysine, arginine, and histidine are positively charged. So we consider electrostatic interactions between these residues. And in addition to that, we have basically total 5 different force fields to account for the many different types of non-bonded and bonded interactions between the different residues. And so in the beginning, we characterized the different structural properties in comparison with experiments, and here I would like to particularly show the results for the radius of gyration, which we find to be quite good agreement with experiments, and even we observe a similar trend as in experiments where the radius of gyration increases with increasing ionic strength. So what happens here is that what happens is that at lower ionic strengths, such kind of self-complexed configuration with contacts between the dipolar N terminal and the negatively charged C terminal are quite prominent, but as the ionic strength increases and the electrostatic interaction gets screened, such kind of configuration becomes less probable, and instead the protein acquires a more extended configuration, which leads into an increased radius of gyration. So from this observation at the single chain level itself, we could anticipate the important role electrostatic interactions may play in the bath solution. So let's see what exactly happens in the bath solution. So these are simulation snapshots for bare alpha synuclein solution. So as we increase the concentration of the protein, so initially at very low concentration, we observe many dispersed protein chains, and we refer to this phase as the fluid phase. But as the concentration increases, aggregates of intermediate sizes emerge in the solution, and finally we observe this large percolated network in our system. And so we basically characterize the size of the different aggregates or self-assembled structures by calculating the cluster size distribution. So particularly for this largest concentration of 10 millimolar, you can see here this distinct peak towards right, which corresponds to the large percolated network that we observe in the system, and it from this plot we can see that it almost contains almost 90 to 95% of all the protein chains in the system. So now that we had some idea about like what kind of assemblies we appear in the solution, so the next question was how are the different protein chains organized, or what is the correlation between the different protein chains. And to answer this, we looked into many different quantities. So one of the quantity we looked at is calculated is the static structure factor, which is accessible in experiments through different scattering measurements. So typically in case of a simple liquid-liquid phase separation or macro phase separation, you would expect the peak for the structure factor to appear at the thermodynamic limit of Q0. But now for the protein chains in the percolated network in our system, we actually find that there is a characteristic peak at a low Q value, which is the characteristic, which is the signature of microphase separation. So actually there is a preference for a characteristic length scale. So to explain it in other words, so basically you have the protein chains organized into small microdomains which are interconnected. So this percolated network is formed of many interconnected microdomains. Then we also looked into the interchain contact map to get an idea about like which residues are interacting with has more interaction with each other, and which is driving the organization into this percolated microstructure. So you can see here that there are a large number of contacts between the N terminal and the C terminal residues. So as we saw in the single chain case, that how electrostatic interactions are quite important determinant of the structure. So here also we actually see that the electrostatic interaction between the dipolar N terminal and the negatively charged C terminal is what is driving the organization into this percolated microstructure. Now, since electrostatic interactions are important, we also did a very detailed characterization of the effect of the electrostatic interactions by varying ionic strengths. So here I want to particularly show you the results for the static structure factor. So here, so what we find is that at lower ionic strengths, when the electrostatic interactions are much more stronger, the peak is slightly shifted towards right. So in other words, the characteristic length scale that appears in the system is smaller at lower ionic strengths, or in other words, these microdomains are much more compactly packed inside at lower ionic strengths. And since alpha-synuclein is very well known as a RNA binding protein, so the next obvious choice for us to characterize this electrostatic interaction was to check how does complexation with RNA affect the structures that we observed in earlier in the Bayer solution. So for this study again, we do consider a coarse grain model for the RNA. Where each nucleotide is described by 3 beats, and we make sure that the coarse graining length scale is consistent with our the coarse grain model for the protein. And again here we have many different interactions to consider, many different force fields to consider the interaction between the different nucleotides. And particularly for the RNA protein interaction, we obviously have the electrostatic interaction between the negatively charged phosphate groups and the charged residues of the protein. And in addition to that, we consider some interaction between the guanine and cytosine nucleotides and the residues of the IDP, because from experiments it is known that alpha-synuclein has a propensity, binding propensity for these 2 nucleotides. That's why we consider these additional interactions. Now with the inclusion of RNA, again, at first we looked into how does the structural property of a single chain looks. So here again you can see the radius of gyration as a function of ionic strength. So in absence of RNA, earlier I showed like how with increasing ionic strength the radius of gyration increase, but now when the protein is complexed with the RNA, we see actually an opposite trend. So now the radius of gyration decreases, increasing with ionic strength. And so for now, if you look into the contact maps, so even though I am showing here the contact map for this particular ionic strength, but for all the ionic strength, you will see that there are almost no contacts between the N terminal and C terminal. So what happens is, so if you look into this snapshot, what happens is that the complexation with the RNA happens through the dipolar N terminal, and the negatively charged C terminal doesn't participate because of strong electrostatic repulsion. Only when the ionic strength is high and this repulsion is screened, this C terminal is able to be more compact and come close to the rest of the body, which leads to a reduction in the radius of gyration. And not just for a single chain, but when we introduced RNA in the bulk solution of the protein, there also we observe a structural reorganization. So in absence of RNA, we earlier saw that how there are large number of contacts between the N terminal and the C terminal. But now when we introduce RNA and increase the percentage of RNA in the solution, these contacts disappear, and instead we see a large amount of contacts between the N terminals in the N terminal residues of the protein. So basically what we find is that RNA is favoring formation of cross-links or contacts between the N terminal residues of the proteins in the bulk solution. And then in this case again to get an idea of the different aggregates or the size of the aggregates that appear in the solution, we calculated the cluster size distribution. So at higher concentrations, we observe the usual percolation peaks, and these percolation peaks actually appear at even earlier concentration with the in presence of RNA. But now at lower concentration, an even more interesting observation appears. So in absence of RNA, this distribution curves were monotonically decreasing. But now when RNA is present in the solution, this distribution curves becomes non-monotonic with a peak at an intermediate cluster size value. So RNA is facilitating formation of small microdomains, in this case even at much lower concentration. So that was basically an interesting observation when we included the RNA. And sorry, so basically, to give you a summary of the main conclusions from this part about this alpha-synuclein IDB solution. So first of all, we saw that at higher concentration, a percolated assembly appears, which has a microstructural organization, and then with the inclusion of RNA, we find that actually RNA reinforces the microphase separation even at a much lower concentration. Now let me go to the last section of this talk, which is the sequence effect, and so this is quite important for many charged macromolecular systems. So initially I mentioned that for IDPs you will quite often see pairs of positive and negatively charged residues close to each other, or even for other charged macromolecules like polyglutamines, for which you will see that each of the repeating unit is explicitly dipolar. Now, to understand the effect of the dipolar interaction, or specifically the effect of dipolar sequence, we considered a coarse-grained model of polydipoles, where each of the monomers were described by 3 beads, out of which the backbone beads were hydrophobic, and the side chain beads could be hydrophilic and could be dipolar. So before going into the effect of sequence, let me share some results about the homopolymer, where basically you have all the all the monomers to be dipolar. So in this case, when we varied the temperature for different combination of the dipole interaction and hydrophobic interaction strength, so by varying the temperature, we could observe the coil to globule transition, and from those transition, when we extract the theta temperature, what we find is that the theta temperature shows a linear dependence on the hydrophobic interaction strength and the dipole interaction strength, and such a p-square dependence on the dipole moment tells us that there can be 2 possible combinations of dipolar alignment, basically the dipoles which are in close contact. But here, so this is a snapshot of the globule that forms at lower temperature. Here you can clearly see that actually antiparallel orientation is the alignment is the main dominant mode of organization for the dipoles, and not just in this single chain level. Actually, when we looked into the gelation of these polydipoles, so this is a typical phase diagram of an associative polymers. So to study the gelation, we constrained ourselves at temperatures above the critical temperature, and so this is a phase diagram of this polydipole system, where you can see this percolation line separating the solution and the gel phase. Now, even inside the gel phase, when we did a characterization of how the dipoles are oriented, so particularly we looked into the angles the dipoles form. So there we see that again there is a preference for the such antiparallel alignment instead of such a parallel alignment with stacking on each other, even though this might have reduced the energy by a factor of 2, but is not but is hindered because of steric interactions, and such a p-square and this p-square dependence observation is particularly important, because typically when you consider mean field theory for this kind of polydipole systems, a randomly oriented dipole is considered, which leads to a p to the power 4 dependence. But what we showed from our simulation is that such a approximation is not always true. So this was for a homopolymer case, but let me actually move to the part which I promised that I will talk about, so which is the sequence effect. And since we are now interested in the effect of sequence, so what we did is that we considered, instead of considering all the bits to be dipolar, we considered only half of them to be dipole and the other half unpolarized, and then we distributed the dipolar repeating units in blocks of the different sizes along the backbone of the polymer. So for example, in the sequence S1, every alternate bit is a dipole, and that's why for our polymer chain with degree of polymerization 256, we have total 256 number of blocks, whereas this sequence S8, in this sequence you have one half dipolar and other half unpolarized. Now for these different sequences, again, when we varied the temperature, we could observe the typical coil to globule transition. But interestingly, in this case, the theta temperature for this transition showed a sequence dependence where this theta temperature monotonically decreased with increasing number of blocks, or in other words, for more homogeneous distribution of the dipoles, the theta temperature was lower. And actually, this is since the theta temperature is correlated with the critical temperature of the system, so this indirectly tells us that actually, so for the more homogeneous sequences, you will observe the liquid-liquid phase separation even at a much you have to be at a much lower temperature to observe the liquid-liquid phase separation. And so we have this such a dependence, but why is it so? To get a feeling about that, we explicitly calculated the free energy difference between the globule and the coil state for each of the sequences. And what we found is that for more blocky sequences, the free energy difference between the globule state and the coil state is large. And that's why to make this jump from the globule to the coil state, you have to be at a higher temperature, and that's why the theta temperature is higher. And actually, this sequence dependence in free energy is inducted by the dipole-dipole interaction energy, and which you can see here. So these are the dipole interaction energy as a function of temperature for the different sequences, and you can clearly see that for S1, which is a more homogeneous sequence, in the globule state, which is at the low temperatures, this dipole-dipole interaction energy is much lower compared to more blocky sequences like S5, and so that's how like this sequence dependence is inducted for the different sequences. And with that, I would also like to basically summarize the two main conclusions from this section, which are, so first of all, for homopolymers, we found that that antiparallel orientation is basically the main determining factor for the structural organization into a gel, as well as for the dilute conformations of this kind of dipolar polymer chain. And then regarding the sequence, we found that if you have a more homogeneous distribution of the dipoles, or not just in dipoles, but in general for any charged macromolecular system, if you have a more homogeneous sequence, it is more probable that the theta temperature will be lower for those kind of sequences. And with that, I would like to acknowledge my collaborators, Professor Muthukumar and Shafang, and also thank you for your attention, and I would be happy to answer your questions.
Hi. Thank you for providing that wonderful insight into synuclein. Just wondering, how would you try and correlate some of these results with its behavior in Lewy bodies in context of the disease?
So actually, in the context of disease, so these are not. So I won't say like these are exactly like amyloid fibrils. So I'm not sure like the whatever we observe in our system, we can directly like comment about the Lewy bodies from basically the condition that we are looking at, that we cannot correlate directly with the Lewy bodies.
But I would like to think that some of this affects the physiology of synuclein in the cell, right? Like its functions in vesicular trafficking, which is what it normally does.
Yeah.
So how does this interface with that? Just some general idea. I understand your focus is different.
Yeah. So
for me to understand as a biologist.
So what is known is particularly for alpha synuclein. So these simulations which we did was at neutral pH. But what is known for alpha synuclein is that if you go to a lower pH, that's when you see that it is much more prominent, much more easier for the amyloid fibrils to be formed. And at neutral pH, it's very difficult.
Hi. I was just thinking that for your RNA and protein interactions, you said that at lower salt concentration, I think you get interaction more with N-terminal.
Yes, yeah.
And then if you go to a higher salt concentration, then in contact map probably you see more interaction with the C-terminal, right?
With the RNA.
RNA.
Yeah.
Right? That's what your observation is.
No, so this is the observation regarding the contact is that when you have RNA, you don't have contacts between the N-terminal and C-terminal, but more contacts between the N-terminals.
Okay, so it is always only with the N-terminal there is contact.
Yes. So what is happening is, if I go back to the, so because since the complexation with RNA happens through the N-terminal, so all the N-terminals can be now closer compared to the C-terminal, and that's why you see a large number of contacts.
So this is precisely my point. The point is that for this kind of RNA protein kind of interactions, people have found re-entrant behavior. That means if you go to higher salt concentration, then might be the one which you are thinking repulsive that can come into picture.
Yeah.
Are you seeing?
So I agree with your point, but for the bulk solution, actually we didn't did a systematic study of the ionic strengths. So that's why. So this is I think so this is at 100 millimolar. So we didn't vary the ionic strength. But I agree with your comment that if I go to the very high salt concentration, I may also see this re-entrant behavior. Yeah.
Protein concentration at which the percolation occurs in your system?
So it will be, if I remember correctly, it's roughly around 6 millimolar or something like that.
Okay. And so the transition from the fluid to the percolated assembly, so this would be a second order phase transition or first order phase transition?
So this is more like a gelation in this case, actually, I would say. So this is a first order phase transition. So for this particular case you are talking about, right? Yeah, no, so this is second order. Yeah.
In the case where you saw non-monogenic behavior, are you proposing that the RNA is causing nucleation and therefore the formation of intermediate sized clusters? Nucleation?
Yeah, so RNA kind of acts like the nucleation center, like basically.
So wherever there is RNA around it, the N-terminus of the synuclein chain,
yes,
the intermediate ionic strengths.
Yeah, but if you look, so for example, so for this one, the 20% one, you can see there are actually two peaks. So there are two peaks. So as I increase the RNA, it's not just the RNA which acts as the nucleation center, but actually also the protein chains also can aggregate.
So, but just to add, this feature that you add a third component and you see the nucleation concentration or CSAT going down is quite well known now. I mean, we see it with DNA and RNA, so it's something, and also in the physics literature, often people call it pre-wetting and so on. So it's so I think with other proteins. Yeah, thank you.